Ladies and gentlemen, I would like to tell you that the one who will be the moderator for this session, her name is Anna Wetterbom, and she is the CEO of the Young Academy of Sweden, an academy for some of the most prominent researchers who are in the transition period between postdoc and becoming a professor. Anna is the mother of two, and she hopes that both of them will enjoy science as much as she does. I think Anna is warm a super high applaud. Welcome, Anna Wetterbom! I'll leave it off to you, Anna.
Thank you very much. Thank you very much, and welcome to this session. With me on stage, I will have this fantastic panel, so please also welcome them. Aaron, Dionne, Malin and Magnus. Thank you very much for being so enthusiastic. As scientists, this is the closest to becoming rock stars we'll ever get, probably. So, something is wrong with that one. I'll get a new one. Okay. Fair enough. Welcome to this session. In this session, we're going to talk a bit about, or not a bit, we're going to talk for almost an hour about scientific method and what it's like to do a science project or research in general. This, of course, varies a little bit depending on your subject and what type of project you're running, but there are some things that are quite common for all research projects, so we will dig into this. And the reason for doing this is, of course, that many of you will, in the next year or next 2 years, do your high school projects. So, has anyone of you already started with that? A few of you. Whereas the rest of you, then you will have a lot to bring home, hopefully, once you start to do it. And we will start, once I get the clicker, I guess, to present our speakers, to show them. They will, one at a time, talk about who they are, where they come from, and say a few words about their research projects. So we'll start with you, Aaron.
All right. Let's see. There we go. Okay, my name is Aaron Coe, and I'm a physicist from the United States. Specifically, I'm from the state of Minnesota, located in the middle and north of the U.S. And I pursued my undergraduate degree at Bethel University, earning degrees in physics and computer science. And now I'm pursuing a PhD in physics at Harvard University in the field of condensed matter. So the research I'm showing here is from my undergraduate career, and I was performing research in the field known as high-energy physics. And physicists want to know what the world is made of and how it works at the smallest and largest scales. So we peer at the smallest particles up to the outer reaches of space. And my project was focused on finding kind of smallish particles using artificial intelligence. Now, you've probably heard of some particles already, if you've heard of atoms, which make up all the matter in the universe. These are formed from protons and neutrons in the center and electrons around them. Now, the particles I want to find are actually bigger than the protons in the atom. And we can find them in nature by using what are called particle accelerators. Basically, what we do is we take two protons and we accelerate them up to near the speed of light and we smash them together. And there's a lot of energy in the movement of these protons, and this can be converted into mass. So we can get something much more massive than these two protons. Now, the particle I'm looking for has been theoretically predicted, but we haven't found it yet. So we want to use particle accelerators. But there's a problem. They're very hard to distinguish from other things we see. So what I do, and what other researchers have done, is simulate what we think these particles look like, and use artificial intelligence to pick them out in the data. Now, my research differs from other similar studies in that it used additional proton collisions, which are present in real particle accelerators. And to think of this, if you look at the image on the left here, this is what other researchers have done. This is what the particle looks like in a particle accelerator, where the colors represent energy intensity, red being more energetic, blue being less energetic. Now, there's some stuff we can learn from it, but the data I'm working with is on the right. This is many more additional proton collisions, and you can see there's a lot more energy. And this is bad, because the thing we want to see is now getting covered up by kind of this fuzz in the data. And so what my research found is that by using effective preprocessing, we could limit the impact of these additional proton collisions. Thank you.
Just click it.
That's you.
No. Oh,
Where's the side button?
right. Now, the stage is yours, Dionne.
Thank you. Hi, I'm Dionne Argyropoulos from the University of Melbourne in Australia. I just finished my Master of Science course, which took 2 years, where I spent 2 years researching how we can use population genetic studies to enhance the surveillance of malaria. So malaria infections can be symptomatic, where individuals present with symptoms, or asymptomatic, where an individual is a carrier with little to no symptoms, but is capable of transmitting disease. These people represent 75% of people with malaria, and so are the focus of my research. And I also conducted a study, or my lab conducted a study in Ghana, in West Africa, which as of last week in the World Malaria Report, is one of the 10 highest burdened countries in the world with malaria. So using a laboratory technique called genotyping, I created a barcode for each parasite in the population. This enabled me to do some computational analysis to figure out how basically the genetic diversity of the population changed over time due to an insecticide intervention. So insecticides, right? They target the mosquito. The mosquito carries the parasite from human to human, so theoretically you would think that the insecticide would decrease the number of infections. And actually that's what we found, which is fantastic. And that's what most studies find, that the insecticide intervention actually decreases the number of infections in a population. But I took this one step further. I looked at the parasite genetic dynamics, and I found that there was still a high genetic diversity over time. This means that parasites are resilient and continue to evade anti-malarial drugs, vaccines, and our immune systems. Thank you.
Thank you. I'll pass it along. Here you are, Malin.
Hi, I'm Malin. I'm from Norway, and I just recently started my bachelor in maths at the University of Oslo. So this project I did as part of finishing high school, and it's kind of based on the idea that plants have certain responses to environmental stimuli. So for example, most plants grow upwards against the pull of gravity. Some plants turn themselves to face the sun, and some plants grow towards water sources. So everything here on Earth is constantly exposed to Earth's magnetic field. So I thought there should be some sort of biological effect if this magnetic field is changed. So there are actually medical devices that produce electromagnetic fields that are used in cases of bone fracture, torn ligaments and tendons, and sprains. And they're said to improve things like substance exchange, oxygen exchange, blood flow, immune system response, and pain reduction. And this is because all cells contain weak diamagnetic, partially charged, charged, and polar substances that would respond to an externally applied magnetic field. So I wanted to test the biological effect of a magnetic field by putting crests to grow either inside this electromagnetic coil, like in the diagram, at a relatively high frequency, or outside. And after a few days, I measured how long the crest had grown and actually found that the crest inside the coil was both longer and straighter. This led me to the conclusion that something was happening inside the cells of the experimental seedlings, which led to either a directional response, so a magnetotropism, or a non-directional response called magnetonasty to an externally applied magnetic field. Thank you.
Thank you. The floor is yours, Magnus. Oops.
Okay, nice to see you all. So I guess I'm the odd one off in the panel, being a bit more senior, representing senior scientist. My name is Magnus Huss, working at the Swedish University of Agricultural Sciences. And I thought I would just say a few words about my journey as a researcher, which all has been about organisms living in the water and mostly about fish, which I've always been interested about. So I started my research training up at Umeå University, Umeå being a city in northern Sweden, where I studied the phenomenon that for most organisms there is substantial variation among individuals, also in the case that they are born at the same time in the same place. So why can that be, and why does it matter? And I did those studies mainly using experiments in ponds and in lakes. After that, I moved on to what you usually do after you have obtained a doctoral degree. I did a postdoc, the first one at the Swedish Board of Fisheries in a small town called Öregrund, where I now used mathematical models to study the dynamics of fish populations in the Baltic Sea, cod, sprat, and herring, as I see in the picture, and their interactions with their prey organisms and with fisheries. Then I moved on to a second postdoc in the US at Yale University, working on implications of both ecological and evolutionary processes on fish and zooplankton, mainly in lakes, working with experiments and with comparative field data. And now I'm back in Sweden at the Swedish University of Agricultural Sciences, working with questions relating to the impact of climate change on aquatic communities for the most part, doing both experiments, observational studies in the field, and mathematical modeling. Thanks.
Thank you. So, how come, Magnus, you chose to dig into fish biology? Why was this your choice?
Well, I guess it's a combination of chance events and the fact that I was really interested in fishing when I was a kid. Yeah.
Right. I'm going to ask the same question for all of you in the panel, why you actually chose your projects. And you've been looking into magnetic fields and how they affect plants. How did you choose it, Malin?
Well, for our project, we had to do it within one of the subjects that we did at school, and one of my favorite was biology, so I chose that. And we were actually learning about plant growth quite a lot, so I already had some knowledge about that. And the magnetic fields was honestly kind of a random idea. Yeah.
Fair enough. What about you, Dionne? Malaria.
Yeah, so that was also by chance. I didn't know really what I was interested in for a lot of my high school and bachelor's degree. So it wasn't until my third year, my final year in my bachelor's degree, where I did a project with my now supervisor, and I fell in love with how you can use genetics, the most microscopic thing, to explain and help us understand a global problem. And also I was inspired by my supervisors, who were all women in STEM. They were all lovely people, and I fell in love with the project as well through that.
Great.
Yeah.
Aaron, quantum computers.
Quantum computers, yeah. So I've been in condensed matter like most of my undergraduate career, and before going to grad school, I thought maybe I should try a different science just to make sure condensed matter really is my field. So I started looking around my university, finding the different projects professors are working on. And I found this kind of high-energy physics project, and I was like, well, it had different aspects to it. It was about fundamental physics and accelerating things up to near speed of light, which I thought was really exciting. And then also had a machine learning and AI perspective, and artificial intelligence is becoming more and more present in science and how we do physics. And I thought that'd be a really great opportunity to learn that skill and then also get to pursue something interesting.
Quite different path to the science you're doing, and I think that's probably the way it usually is. And particularly when you're going to choose high school projects, it might be random sometimes. When I did my high school project a number of years ago, I had no idea what I wanted to do. I was doing science, and one of my best friends, she didn't have any idea either. She was doing technology, but then a school called into our high school. So it was a primary school calling and said, "Oh, we have this project where we want to." We would like female students to come out and teach our students, and they were like 9, 10, about computers. Right, and this was in the 1990s, I can confess, and so computers weren't that common in classrooms. It's unbelievable today, but and my best friend Maria said, "Oh, that's a cool idea. Do you want to join?" I was like, "Yeah, great. Then we have a project." And looking back at it, I don't know how we got it actually passed, because we did. I mean, we were basically doing community outreach and documenting it. We weren't really doing any research. We weren't testing things with different groups. We weren't testing a hypothesis. We weren't doing any experiments. It was more of an outreach project for younger kids to show them that computers can be fun and sort of yeah. Probably you wouldn't do that today, because nowadays teachers and parents don't want to show the kids anymore that computers are fun. You want them to do other things. But that's what I did, and I passed that one, obviously, because then I went on to university and I studied engineering. And I was completely... I had decided that I was going into industry. I was doing biotechnology engineering, and I thought, well, I'm going to go into the pharmaceutical industry. Then, at the end of my education at university, you had to go out and do a project, it was compulsory, so somewhere. And I went to the pharmaceutical industry, to AstraZeneca, and I was told by everyone, well, if you want to do science in our industry, you have to go back and do your PhD. So I did, and then I spent a number of years working in science, in biology, but and I do like biology. It's not like that, but I liked programming much more. So I was more of a coding person. I tried to stay out of the lab, unlike you. So path to science can look quite different. And now I actually don't work with science, but I work a lot with scientists. So there are many paths, and there are many choices, and sometimes they can be quite random. And that's fine as well. So, now we have got to know you. Now we're going to talk about actually the timeline of a scientific project. Isn't that exciting? Yes, it is, because this is actually what you're going to follow through when you do your research projects at the end of year 3. So, we're going to walk you through this with the help of the panel. We're going to talk about the different phases here. And we're going to start from the very, very beginning with the pre-study phase. So I'm going to turn to you, Aaron and Dionne. First, for you in your projects, what did this include? You know, pre-study, what is that?
Want me to go first? Yeah, so just so you all know, you already started it.
Yeah.
In your classes, when you're learning material that you're probably going to be applying in your research. And this can sometimes not be too clear. I've been in classes before where I've seen materials, and one of them is like what's called a frequency comb. Basically, if you play an instrument, there are different notes, and these correspond to different frequencies. And if you space these evenly, it kind of looks like a comb, so that's what we call it. But when I saw this kind of concept in class, I thought, I'll never do that. But I still kind of paid attention a little bit. And then like a year later, I ended up doing that project. And I was like, well, I'm glad I paid attention to add notes on this. So you should really be prepared and be willing to take notes. And then also your other research projects will lead to additional information, additional techniques, and kind of how you think about problems. A lot of what you're going to be learning in school is how you think about problems and how you approach them. And then probably lastly is doing all the research, which can be kind of thrilling and kind of boring at the same time, because scientific papers are very, very complex sometimes and very dense. But the more you read, the better you get at reading them.
Yeah, I'm going to echo that. I do a lot of reading, a lot of pre-reading, and I keep it up. Even when you're doing your experiments, even if you're almost in the stage of writing your thesis, you should still be reading, still doing the pre-phase. The pre-phase is kind of always there. You always are in that development stage because you're always learning new techniques. That's also the really exciting thing about being a scientist. You're always learning, you're being curious. So I read a lot. I also try to... it takes the longest time to read, as you've kind of noticed, but to take bits of papers, asking your supervisors for help, those kinds of things are really what starts you off on the journey. Yeah.
Right, so I understand there's a lot of reading here to understand learning things, understand what other people have done before you and how you can use it in your own project.
Yeah.
But how do you know what to read, and how do you know what you can trust?
Well, that's a really very valid question. You have to find what's valid and invalid research and what's good and bad, and the only thing you can do is basically, I guess, go to Google Scholar, right? Type in a search word and read it. If you like it, look at the references in that paper and continue reading. Go down a little bit of a rabbit hole. It's actually really fun. Again, also talk to your supervisors. They can help you start in that direction if you're in that position to have a supervisor. If you're not, again, just there's a lot of apps on the internet as well. There's one called Researcher you can put some keywords in. You can find whatever kind of title that sparks your interest, and you read that paper. So that's one way to get the ball rolling.
Yeah. I would also say Google Scholar is amazing for finding research.
What is Google Scholar?
Google Scholar, like if you've used the Google search engine, if you type things, it comes up with results. But if you type Google Scholar, Google will take you to its kind of scholar section. Basically, when you type in keywords, instead of bringing up kind of different websites, .coms or .orgs, it'll bring up papers for you to read, scientifically published papers. And this is nice because publication can help kind of give a credit to the paper. If you see it's published in different journals, like very prestigious journals. Or you can look and see like where the authors are from. If they're in their basement or if they're at a university studying with grant money from a government, then that's a little bit more of credit to them. But yeah, it can be hard. And even in the scientific community, you can have publications that directly contradict one another. And you kind of have to decide which one you're going to believe. And as Dionne has said, talking with other people in your lab and talking with other researchers can help you kind of figure out who you're going to be supporting in these cases and who you're going to believe.
Yeah, I'm going to just extend that a little bit as well by saying that when you're reading these papers, it's important to critically read them. That means don't take it by face value at the start. They might use different methods to what you're considering, or they might have, I don't know, that peer-reviewed papers are really important, and that's what you usually find on Google Scholar. There's a little symbol to let you know that this has been read by various scientists. So keeping those little things in mind can help you get on the right track.
Right. I heard in the introduction that I had obviously written that I'm a mother too, which is true. My kids are in 5th and 7th year of school, so they're 11 and 13. And I have noticed that my kids learn a lot about how to be critical to sources, which is of course good, but no one has taught them really what to trust. And so I think this is a key thing, what can you trust? And you mentioned peer-reviewed papers, and that is when scientists write down what they have done, all the results, and why did this happen, and what do I think this means. They write an article, and they publish it in a scientific journal. But before they can publish it, it's sent to other scientists who will do their best to tear it apart and criticize it to really, really make sure it's worthwhile, it's trusted, the science is good and solid. So once it is actually published, it is to the best of the current knowledge. Then things might change over time, of course. And that's the difference, why scientific journals and scientific papers are solid in that sense compared to when, if you would write a blog post, for example, no one would check that beforehand. So I would trust you less when you write a blog post or Twitter or whatever social media you use than if you actually publish a paper. So this is a tricky part, and I'm sure you're going to talk about this before moving into your projects. So there's been a lot of reading up until now. Preparations then, apart from reading. Both you and Malin, you and Magnus, you have worked with experimental projects. So how do you prepare for that, for the experimental studies, before you actually go into the lab or go out and start fishing? So what do you do to prepare?
Well, I always start with, and that irrespective if you're doing experiments or something else, is that you need a question.
Yeah.
And that really is the first thing. And you should remind yourself over and over again, what is my question? And maybe you will rephrase the question as you go along until you start experiment after reading all those papers. And that question will guide what you're supposed to do and how you design your experiment. So once you have that question written down and have talked it over and over again with colleagues, advisors, if you have any, and so on, you start to design the experiment. And then there's really two parts. There's this sort of theoretical part where you set up your experiment, trying to figure out what experimental units and so on to have. And then there's, at least in my case, also a logistic part. Can I make this work? Can I go out in the ocean and put up some bags without them blowing away to Finland? Maybe, maybe not. So there's those two parts, and both need quite a lot of preparation, including backup plans.
Right. Malin, what about you?
Well, I really like to figure out all of the control variables I need to have in my experiment that could actually interfere and kind of mess with what I'm actually trying to find out. So yeah, going on with that, you should have a very clear idea of what you want to do and how you want to find that so like what you want to measure, for example. And also maybe figure out your criteria for what you need to be able to accept your hypothesis. So for example, if you do like any statistical tests, you should be clear at the start like what significance level you can accept.
Right. And these two phases, talking about preparations and pre-study, that's usually the part you sort of kind of forget about when you think about scientific projects. You usually think about, we're going to run all these cool experiments and go out in the field and study things, or sort of find new things. But these are essential, because if you don't prepare, you might end up standing in the lab and then realizing, I forgot to order these things, or I forgot to start growing my plants on time, and you will have to wait for a couple of days, a couple of months, depending on what you're actually after. So it's maybe not as exciting as the rest of the process, but it can nevertheless be sort of the showstoppers if you don't do them, right? With that said, now, sort of read up, you know what you're going to do, you have the hypothesis, you have the material at hand, you might even have built things like I know you do, Aaron. Yeah. Now, it's time to run some experiments, right? And I'm going to ask you again, Malin and Aaron, for you, how did you plan and perform the experiments in your projects? You want to go first?
Oh, well, I like to write out my method first, because my experiment went over a few days, so I had to make sure that I did pretty much the same thing every day so that nothing was going wrong. And then when performing every day, I tried to check all the control conditions I had to have. So for example, I checked temperature, humidity, and the amount of water I was giving my plants. And I was also trying to be, again, so that everything was the same every day. I also tried to stick to certain times when I would, for example, water the plants or turn on the magnetic field, and then obviously gather results. Yeah.
Right, and you said I write my methods, and when you write your scientific report, you always have to have at least a paragraph explaining the methods, what you've been doing. And you're right, writing that down beforehand can be quite useful, because then you know what you're going to do. Because the opposite would be to write it afterwards, and then you usually realize, well, I forgot about this step, and I forgot about that, and oh, why didn't I do this? And then you have to go back, right? And what about you, Aaron?
Yeah, so kind of echoing what Marlon said, having a very concrete process to go through and have a thought-out process, it's really important to be able to kind of replicate your experiments. It's kind of terrible if you end up changing too many variables at once and you get results and you realize you can't distinguish what happened or why it happened. Or if you didn't write down what your process was and you got these amazing results, but you have no idea how to get them again. And kind of writing down in the lab notebook. Notebooks are extremely important. I think we'll talk a little bit about when you're writing your papers. It's really important to take good notes, know what you've done and what the experiment did, so you can communicate your results with other people.
You're completely right, and that's usually the boring part. At least it was to me to write, but I've learned the hard way that it's good, otherwise you have to redo. Mm-hmm. Doing experiments, my experience at least is that it doesn't always turn out the way you expected. Sometimes it didn't work at all. Sometimes it was a bit unexpected. Has that ever happened to you?
Yeah, I mean, you get results. I mean, with the research project I was showing earlier, you know, we got kind of confusing results at the end, and sometimes it can be a good thing to get weird results. I mean, you know, usually you have an idea of what's going to happen in your experiment, and then at the end, when you get it, you're like, oh, this is pretty cool. I got what I wanted. It's kind of like having a great Christmas gift or birthday gift. And then sometimes you get something that, you know, it's not very extraordinary, and it really means your research wasn't, you know, as much as you wanted. But sometimes you can get a result that doesn't make any sense whatsoever, and it's completely different from what you expected. Maybe you've heard of superconductors before. Basically, you know, the wires you plug into the wall carry electrical current. But there's some kind of like resistance of this electrical current, like if you're pushing on water down a stream. And in superconductors, it falls to zero, which is really great. And it's a really cool process, and we talk about them a lot in condensed matter, and it's a huge field, and it has so much grant money from so many governments involved. But the first guy to discover it didn't think much of it. He didn't even notice it. He didn't expect it. And so sometimes these weird results that you don't expect can actually be quite extraordinary.
Mm. Right. And what about you, Malin? Did all your plants grow as expected?
Well, most of mine did, actually, so I was very lucky. Yeah, as you said, like a gift. But yeah, I guess if something goes wrong, like you said, you shouldn't be too quick to throw them away, like the results you don't want, because you might actually want them because you might be coming onto something new. But I guess maybe you should also think about how your method went. Maybe there was something wrong in the method, or maybe like something was interfering with your experiment that you hadn't considered before.
Right. So having collected your data, you have some preliminary results, or at least you have something in your computer. Then you run your backup because you don't want to experience, tipping out the coffee or the cola or whatever you drink on your computer. So many people have done this. Don't do it. I'm married to an IT consultant. That helps. But for the rest of you, run the backups at this stage, okay? Unless your parents are IT consultants, then you're fine. So now we have the data in a safe place. It's time to analyze it, to find out what actually happened. So Dionne and Magnus, let's start with you. What do you do when you sit down, and how do you start, basically?
Yeah, it's overwhelming to start because you've done all these experiments for so long, and you're like, yes, I can actually get into it. It's exciting. So to get started, I think you have to be responsible. You have to make sure all the numbers are there. Numbers are really important, especially with data, to make sure that you've accurately counted as many, well for me, the amount of parasites and humans that were in our sampling and all that. So making sure that the numbers are all correct. And then making sure, I guess, you keep your goals in mind. So what that means is asking the question that you're interested in. And when you're analyzing the data, you don't stray from that. And that's very easy to do. You could always find an answer for another question. But it's just really important that when you start getting and sorting through all that data, you write down a few questions, things that you really want to find out from all of this mass that you've got, and you stick to it. And then you can change from there, I guess. But it's good to start. Yeah.
Yeah, I very much agree. I think it's, again, really important to remember your questions and try to analyze the data in a way that you can answer those questions. It's very common that you do get results that are super interesting and maybe warrant a second paper or something, but it might not have anything to do with the question that you want to answer right now. And then, of course, it might be difficult to disregard those results for now, but sometimes you should. And then I often start out as soon as sort of the data is quality checked and in some sort of database or so to visualize the data early on before I do statistics on them to really see, so what does it look like? And often within those 10 seconds when you create that first graph, you have actually a quite good grasp of did it go well or not. And sometimes, of course, it's immediate, it went well. Sometimes, no, it didn't go well in the sense that I hope, but then it's often still possible to find out that it actually went well in the sense that you found other interesting results. As Aaron said, you can also find unexpected things in your data. So I guess that's how I would start.
Right. So when you visualize the data, as you said, you make a graph or sort of to see what happened. You often get these annoying little dots that don't really fit. Can you just get rid of them?
No.
Okay.
And I work with biological data, and often in not super controlled environments, often outdoors, for example, and then they are not well fitted on a line ever. And those outliers, that sometimes they are outliers in a statistical sense, and then it's okay to treat them as outlier when you analyze the data, but that doesn't mean removing them so that the readers can't see them. So I think that's in general very important. Inform the readers what do the data look like, and then you can choose to do statistics. There might be rationales for why you can remove certain data points when you do your statistical tests, but then you first have to test that it is true outliers that should be removed and not. But my suggestion is that you always show the data that you have.
Right. Since you are supervising people, I will turn to you, Dionne. Well, I mean, sometimes it happens when you're a student and also when you're a more senior scientist that you made a mistake in the lab, and then you get the results, and then you realize, okay, I swapped the samples, I did something completely wrong here, I spitted on the glass while I was going to do microscopy, and it looks odd. Do I have to tell my supervisor, or can I just sort of quiet it down?
You have to tell your supervisor. Your supervisor, they're there to actually help you. So yes, your pride, yeah, sure. Just put it aside a bit and accept and be honest. Honesty is integral to your, integrity as a scientist. It helps you grow. So yes, you have to tell your supervisors. It's happened to me. I did a year's worth of work on this really cool data, and I was so excited. And then I realized that one of the basic fundamental things I should have done at the start to analyze all my data, put it through a program. I put all my data through at different times in the program rather at the same time. And that actually gave me different results. I won't go into the specifics, but it turned out that this second realization that I came to was really integral to what I got as results. So I bit the bullet, just went up to my supervisors and said, look, I'm sorry, I did this, and they were so lovely. They let me have my time to get everything back to normal again, and it was all fine. They actually commended me for being honest at the end of the day. Yeah.
It's usually the case. If I'm going to share one of my mistakes when I was doing science at graduate school, we were looking into the DNA, the full genome of 2 chimpanzee samples. One was female and one was male. And chimpanzees are like human. They have this, in the sense that the female would have 2 X chromosomes and the male would have X and Y instead. And then we decided to plot the genes on different chromosomes, and it turned out the chromosomes didn't fit with what we thought was the male and the female. So somewhere along the lines, me and my collaborator must have swapped the samples. We'd been working on this for a year, then we got the data, and we also went actually to our supervisor, and she was working also in the clinic at the hospital as a geneticist, and she just looked at us and said, well, no one died. And she was right, no one died, except me and Adam, perhaps from having to admit this, but we got over that, and we actually wrote the report on it in the end. So I think it was, we learned something from it. And you also told me when we talked before, Aaron, about your friend who had been doing all this science and then decided to cut out some of the data.
Yeah, well, I want to talk about another story. There could be even more extreme examples of like what can go wrong in the lab, and you can feel embarrassed about it. And I want to come to your supervisor. Like, I've had friends who accidentally built bombs in their lab, and then they have to go to their supervisor and be like, I accidentally built a bomb, I'm so sorry. But you have to, because if you don't and it goes off, it'll blow up. So, and like, no one died because they quarantined the room, so it was good. But
how come they accidentally build a bomb?
it's easier than you think.
Okay, maybe we shouldn't go into that.
Yeah, we won't explain that on stage. But yeah, I did have a friend who was plotting some experimental data, and he often in physics you can have kind of theory and then you have experiment, and sometimes you want to see if the experiment fits the theory or the theory fits the experiment. And he had been taking these results, and he found that in like a certain area of his data it fit really well with the theory, but then the other areas it was really off. And so when he was presenting his paper, he just didn't show these areas. He only showed the range of which the experiment he believed worked. And he got asked like why he kind of cut the plot off on these sides, and he said, well, it didn't fit so well, so we don't know if something's wrong with the experiment or not. And they found out a few years later when he went back to the lab that the theory actually turned out to be wrong, and his experiment was evidence for that. And if he had brought it up earlier, he probably could have corrected the theory. So always kind of try to be more honest with your results and kind of come forward, and don't be too embarrassed or feeling that someone else must be right because they said it first.
True. It's the unexpected results that sometimes, at least, you find the new science, the things that you didn't know that you were even looking for. And in this Nobel week, a lot of actually Nobel prizes come from these spurious ideas and sort of results that you were trying to do something and then it turns out completely different. It looks in the process as it's linear. It's not at all, actually, because this redo phase that we put in here is quite common, that you run your experiments, you look at the results, and you realize you have to do a lot of more experiments to figure out what's really going on here. So, Malin and Dionne, How common is that you have to redo things, Malin?
I think it's pretty common, especially like you want enough data to analyze, and you want to kind of get rid of the effect, because there's always like human error involved. So the more trials you do, the kind of less effect those have. So for example, I had like 100 crests in each condition, so 200 crests overall. So plants that I measured the length of, and that kind of gave me a good amount of data to analyze. Yeah.
Yeah, well, science is built upon replication, replication of your data, replication, just to make sure if you're not quite content or happy with how something went because of human error. For example, if you slip in the lab or you pipette something in the wrong little area and you're not sure, write it down in your lab book. That's why it's good to have good lab books. And then you can always go back, do that, redo that sample, find the data, and then continue on. It's always good as well. For the nature of my work, I have to make sure that I don't do too many mistakes. And, you know, you're human, right? It's human to make mistakes. So when you're doing your experiments, it's really important to take time. Don't ever rush things. If you've got commitments or you want to make plans after you're in the lab or whatnot, make sure you give yourself time to pack up and do everything properly, because you really don't want to rush your experiment just to go to the cinema, really. It's not worth it at the end of the day, so yeah.
That's why I never did much lab work, because I don't have the patience. So I went over to programming instead, because it's much cheaper when you fail, usually. It's only my time. It's not a lot of expensive equipment or material and chemicals. Well, once you've redone the things that didn't work out, and you have redone things to figure out what actually happened, finally it's time to sit down and write. You know, how do you feel about the writing, Aaron?
Well, it's a bit of a roller coaster, actually. So initially when I'm writing, I'm kind of excited because I got these results that I want to put out into the world and communicate with other people. So I'm really excited about it. I type away. And at first it's like, whoa, this is pretty cool. And then after a while, I'm kind of bookkeeping, making sure I mention everything, and that's not so much fun. But it's a process you have to go through. And we talked about peer review earlier. This is when you kind of send out other people review your data, review your paper, and then give you comments and feedback on how to improve it. And this process can be kind of long and arduous, but it's a good thing to go through to become a better writer and a better communicator.
Malin?
I like certain parts of the writing phase. So, for example, like the introduction part, I find quite fun because you actually get to formulate your idea and get it on paper. And there's, I guess, some parts that are tedious but still very important, like the method part and explaining your experiment setup. Can be quite a lot of like repetition and being very specific about details. But I guess at the end, once you're done, it's very satisfying because it's like your work and it's your own thing that you've kind of finished and come to an end with.
And the more time you put into it, the better it looks. It's like,
It's a nice feeling, you know, when it's in front of you. Magnus, you've been writing quite a lot, I guess, over the years. Do you have any advice for people in the panel or for the audience when you get to this stage?
Yeah, I mean, it's very difficult to give general advice, and sometimes I do give advice often on how to write, but it's not always the way I would do it myself. So when I give advice, I would say that, make sure you have an outline, which could be a graphical sort of abstract or bullet points. But if I'm to be perfectly honest, I often just start to write. If I'm in the mood for writing, given that you need to know the question and have the answer. That's the bare minimum, else it's impossible, I would say. And often that's the problem with writing. It's to get started and keep going without worrying of every comma in your text. And I would say it's maybe the part of doing science that limits the most people from finishing. It's the writing. It's not doing the statistics or setting up the fancy experiments. It's to write it up. So I think it's super important to work a lot on writing, and it's a skill. So everyone can practice and get better. You're not born a good writer. It's a skill that you gain from practicing.
I think what you said on just get started is perhaps the most important thing. I'm not good at starting writing, but to sort of ease my own nerves, I try to write down some headings usually, and perhaps put in a few words every here and there, just on what I'm planning to write in the different sections. And then I just close that document and then come back a week later. And then I open and say, I already started. So you try to sort of basically fooling yourself in thinking that, I'm onto this, and what you weren't really. But once you start writing, you actually get there.
Yeah, and I really liked what you said about not putting every comma in, because usually when I'm writing, if there's like something I'm not too clear about or something I really don't want to write at that moment, I usually just put the word blank in capital letters. And this is really good for me to just like get beyond that and just write the things I want to get down and make progress. Of course, I have had instances where I gave the paper to someone else and forgot a blank, and then they're like, "Why'd you write blank on your paper?"
Yeah, well, you write first and then you edit later.
Yeah.
And also there's a technique called the Pomodoro technique. It's basically where you just write for 20 to 25 minutes, no phones or anything, it's just you and the computer. You write, don't really, just get brain to computer to fingers, get it all out there, and then you can edit it later. And you do that with a 5-minute break, and you keep that cycle for a little bit, and then eventually you can edit it all later the next day. But at least you get it out of your mind.
And you can take fika during that 5-minute break.
Yeah, exactly.
Yes, and a lot of fika. You're right. Coffee drives science, if you haven't already discovered that. Now, looking back then at the whole process of a scientific project, which part do you enjoy the most? Let's start with you, Malin.
Well, in my project, I think I enjoyed the data analysis the most, because I had both qualitative and quantitative data. So like I had the lengths, but I also had pictures of the crest, so I could kind of use both the types of data to work together to kind of formulate a conclusion. So that was pretty cool and fun.
Yeah, well, every stage has its own merits, but I completely agree. Data analysis is the most exciting, because you get to piece the puzzle together, you get really make the graphs, and you're like, I know where that is. It's really cool. So I like that for sure. You become a detective and you're piecing things together. It's very exciting.
So I'm a little bit torn between whether or not it's data analysis or the experiment. I mean, they're both really fun to me, and I guess the experiment is almost like a birthday present. Like, you get this box, and you're not sure what's in it. You can kind of shake it to see what, but you can't really
bomb?
guess. It's not, hopefully not a bomb, but like, you get this box. You got me there. So.
Sorry.
Oh, no. That would make data analysis more interesting, but so. I guess the experiment's like the present unopened, and then with data analysis, it's kind of like opening the present. And this can be kind of a mixed experience. You can either find like a PlayStation, which is really cool, or your grandmother got you socks, and that's nice, but it's not as cool. Or maybe get something totally unexpected, and that could be really awesome too. You never know what you'll find, so I'm kind of torn between the two.
Right, Magnus.
Yeah, I think maybe I differ a little bit here. I think, well, first of all, getting sort of the idea in your head formulated, that is really fun, to think up something cool that you want to answer to. If it's running an experiment, especially if it's sort of a field experiment, maybe then the first day out, starting it, that's really fun. Then after a while, it's more a lot of work. Then the first time visualizing the data, press enter, okay, maybe it worked, maybe not. And then often I actually think starting writing it up, because quite often that's sort of when I realize what it actually was about somehow. You might have formulated a question and written down some methods, but when you start writing the introduction and discussion, it's sort of then maybe sometimes it's actually you realize this was much cooler than I actually thought it was, and it fits this big picture. Different parts.
Right, going through all this sometimes frustrating and also quite time-consuming process of working through a scientific project. What is really your motivation? Why do you love it enough to do it?
Yeah, it's sort of difficult to say. I really like working with organisms, with nature. That's sort of a genuine interest in itself. Then it doesn't have to be about fish. I happen to be interested in fish, but I could just as well have worked with lots of other organisms. So I think it's more about figuring things out. And it doesn't have to be a specific question. Then after a while you sort of drift into getting into a subject, and then you just get really interested in this specific. I really work a lot on fish, and how they are size structure and how that matters for how they vary in time and space. But if I would have started working on fruit flies, I would have been super interested in fruit flies or whatever, I'm sure. So I think it's just start to dig into something that you initially find interesting, and then you will end up being even more interested in that subject, I'm pretty sure.
Right, and what about the rest of you? Why do you love science? Malin?
I like it because it's like a conclusion to an experiment can kind of lead to a new experiment and a new conclusion and so on and so on. And then you have this theory, but then people try to like test this theory and try to disprove it to get to another theory. So it's like this never-ending cycle, which is really fascinating. And then also science makes reality seem pretty cool as well.
Yeah, curiosity. I love science because it embodies curiosity. It's always asking the questions. It's about finding answers, and answers usually lead to more questions, and you kind of just keep going and going and going. So that's why I love it. It's like you're a child every day. You get to ask all these questions with big eyes. You're like, ah! Yeah, that's why I love science, for sure.
Right.
Yeah, I like to echo that curiosity and talk more about a kind of a personal young story. So as a kid, I really wanted to fly. I thought that was the coolest thing ever. And, I kind of looked at TV to find the answer of, like, how you fly. And so, like, I tried the Superman pose, didn't work. I tried just believing I could, that didn't work either. I tried running, starting, copying the birds, that also didn't work. And so I remember out of frustration, I came to my mother and I said, well, I can't fly. And she was kind of puzzled, like, yeah, of course. But then she decided to buy me a remote-controlled airplane as a kid. And I had seen airplanes on TV, but nothing else on TV worked, so I didn't really expect this to work either. And then kind of like building and putting together, and then suddenly getting to fly it. And as a kid, I just kind of stood in awe as this thing was gliding through the air. And I remember at that point, like, realizing whatever made this plane fly is what I wanted to study, because I realized this mechanism allows you to do extraordinary things.
Thank you so much for sharing your love of science and how you work through a project. You will have the opportunity to talk to them outside of the poster session later. Please do that. And thank you so much in the audience.
Amazing, amazing. High five.